Hi everyone, we're going to review the autoimmunity 1 and 2 lectures. For your convenience, I've combined the 2 lectures for the review session since they're on the same topic. So here we go. The major features of our immune responses that are able to reduce our risk of an autoimmune response are basically stimulating self-tolerance. So that can be through some passive mechanisms and it can be through some active mechanisms. But I will briefly remind you that central tolerance is a tolerance mechanism that deletes or edits self-reactive T and B cells, those that are too self-reactive. Antigen segregation refers to the fact that those T and B cells may not be able to actually directly access their antigen due to a physical barrier. So you can think of the central nervous system, for example. Peripheral energy is about the fact that cells are getting a weak activation signal because they're not getting co-stimulation. So they don't make a good response after that kind of activation. There are more active mechanisms such as regulatory T cells, which can suppress directly the responses of other cells. There's functional deviation where cells acquire a different phenotype than they typically would under normal activation signals. And then, of course, there's basic apoptosis activated cell death. We talked about the fact that both genetic and environmental factors could lead to the development of an autoimmune response. What they share is that they can both permit immune dysregulation and that this ultimately leads to chronic inflammation and autoimmunity. The strongest example that we talked about of a genetic influence on the risk of autoimmune disease development is the human leukocyte antigen, or HLA locus, in the genome. We talked about what a risk allele means and the relative risk, what those mean, but to briefly cover this, for the example of ankylosing spondylitis, the risk allele is called B27, and the relative risk is quite high at 87. And so what this means is that people who have ankylosing spondylitis are basically 87 times more likely to have the B27 allele than controls. This does not mean that everybody who has the B27 allele has ankylosing spondylitis, though. We talked about different categories of hypersensitivity responses in the context of autoimmune diseases, and so I will go over a few of those briefly to remind you of the mechanisms that are involved for each of them. So we talked about type 2 autoimmune responses. This is where antibodies are produced against cell surface or extracellular matrix proteins. There's a list of different examples here, but let's just go straight to one example. One example is autoimmune hemolytic anemia. So this is a type 2 hypersensitivity response because IgG or IgM antibodies are actually specifically binding surface proteins on the surface of red blood cells. So when those bind, they can interact with Fc receptors on phagocytic cells, or they can activate complement receptors on phagocytic cells. And ultimately, this leads to destruction of the affected cell. So over and over again, you will see the same pattern where antibodies are binding a surface or an extracellular matrix protein, and that ultimately leads to tissue destruction. We talked about some type 3 responses. Type 3 responses are characterized by immune complexes. The classic examples are things like rheumatoid arthritis and lupus. In rheumatoid arthritis, there are autoreactive CD4 T cells that stimulate antibody-producing B cells to produce rheumatoid factor-specific antibodies, as well as antibodies to citrullinated protein antigens, or ACPAs. So basically, rheumatoid factor is actually IgM antibodies, and they're specific for the constant region of IgG antibodies. So don't worry too much about the details of this. The point is that you can detect rheumatoid factor antibodies and antibodies to citrullinated protein antigens and that those antibodies lead to immune complex development and ultimately lead to cartilage and bone erosion. In lupus, there are many different mechanisms that are involved, but a major one is that programmed cell death is disrupted. So apoptosis is affected. So normally when a cell undergoes apoptosis, this is kind of quiet death. And so the intracellular components of those cells are hidden away from your immune response. But what happens in some cases of lupus is that this death is not quiet. And so your antibodies can actually see these antigens and respond to them and form immune complexes. We talked about the fact that certain patients and mouse models have a mutation in the FAST gene, which is very important for programmed cell death or apoptosis. And so if this FAST gene pathway is affected, you can actually drive an autoimmune pathology that looks like lupus. We also discussed some type 4 type responses. So these are the T cell mediated autoimmune diseases. And you can see that there are many different flavors of this. We talked about several of them in detail, but let's go over a couple to remind you. So one example that we talked about in quite a bit of detail is psoriasis. This is a Th17 typically mediated response. It's characterized by skin inflammation. Plaque type responses on the surface is what is most common. The big thing, though, is that ustekinumab is one of the common treatments for psoriasis. The way that it works is very important. So basically, it's able to inhibit both Th1 and Th17 responses because the antibody is capable of binding the P40 subunit of both cytokines. So both IL-12 and IL-23 can be blocked. And so those correspond to reduction in both Th1 and Th17 responses. In celiac disease, typically the antigen that we're talking about is the immune recognition of gluten, which is of course a staple in many people's diets. There's a strong genetic component to celiac disease. So 95% of patients who have celiac disease have this HLA type called DQ2. We know that there's a strong genetic component because there's 80% concordance between identical twins. But basically the way that this works is that peptides are produced from gluten. Under normal circumstances, those peptides don't bind to MHC class 2, DQ2. However, when these proteins get modified by an enzyme, transglutaminase, that can lead to modified peptides that can, in the case of people with celiac disease, be presented on MHC class 2. This bound peptide then activates gluten-specific CD4 T cells, as you can see here. And then those activated T cells can ultimately lead to killing of the nearby epithelial cells. So I don't want to go into all the details of every autoimmune disease we talked about for the review, but in principle, you should understand the different types of hypersensitivities and how those relate to the autoimmune context as well. So we did talk about a variety of different treatments and biologics that are used for the treatment of autoimmune diseases. So an important one for you is to understand how monoclonal antibodies are named, because that tells you something about the type of antibody that's being produced. So recall that fully mouse antibodies end with this OMAB ending. Chimeric antibodies are ZMAB. So for example, infliximab. We have Zumab for humanized antibodies, where there's a small number of mouse components left. And then we have the UMABs, which are the fully human ones, such as adalimumab. So this is the generic drug names that are regulated by the FDA. As I mentioned, they can tell you about the type of antibody that's being used. And in general, as you might imagine, something that is less human is more likely to be immunogenic. We talked about some other possible targets for the treatment of hypersensitivities in autoimmune diseases. These can involve targeting or blocking cytokine and cytokine receptor interactions. They come in all kinds of different flavors. So of course, they tend to be targeted to a particular family that's involved in a given autoimmune disease. They can look something like this. In this case, we're looking at a decoy receptor type example. So if a patient has a high level of a certain inflammatory cytokine in their blood, those cytokines can bind the receptor on the surface of their immune cell and activate that cell. However, with the treatment with the decoy receptor comes in and actually mops up all of this residual cytokine. So they're all bound up by the decoy receptor, and so there's none left to bind to the immune cell, and thus the immune cell does not get activated. So that's, of course, an attractive drug target because it can be very specific. We also talked about different forms of TNF inhibition. TNF is a central cytokine made by macrophages and Th1 T cells, among other cells as well. These are the two primary candidates for production of TNF. And basically, modulating TNF leads to modulation of inflammation. And so being able to change the level of TNF activity is really important in the treatment of autoimmune and other diseases. It comes in lots of different flavors, though. So there are some that target using an antibody, which binds it up. You can have decoy receptors that basically pretend like they're TNF receptor and block the ability of TNF to actually bind up the response. And we talked about what those structures can look like. So as you can see here, etanercept is the brand name for this. We see a decoy receptor here that is capable of binding TNF, and that's attached to an Fc component just to keep it stable. And then we have actual antibodies where the variable region of the antibody is capable of binding the TNF cytokine itself. So these types of drugs are examples of biologics. We talked about differences between small molecules and biologics. You should have a fundamental understanding of how that works. And I will stop there. Please feel free to reach out if you have any questions.